Right. Now, three barriers are there: corona radiata, zona pellucida, and vitelline membrane. Clear? Now, I already told you that these corona radiata cells held together by hyaluronic acid. Right? Now, all these sperms start producing one substance called hyaluronidase. What is it? Hyaluronidase. 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 What is the function of it? It is agonist to the hyaluronic acid. That means it will digest the hyaluronic acid. Clear? When it digests the hyaluronic acid, what will happen? Bonding between these cells will be removed, so that these cells are loosely arranged. When these cells are loosely arranged, sperm will reach here and movements of tail, flagellar movements. But flagellar movements means what? Bending and stretching of tail. That means propulsive movements. When this propulsive movement is happened, this head of sperm will hit these cells. When it hits these cells will disperse. That means they will give space for the passage of sperm. Clear? So, now this sperm reached here. That means it distracted one barrier and reached to the second barrier. Clear? So, now the sperm is here. Now the sperm is here. Right? Now the sperm is here. Now when it reaches to the zona pellucida contains some receptors called ZP2, ZP3 receptors. These ZP2, ZP3 receptors receives the sperm. Right? That means sperm will go and attaches to these receptors. Right? Once it go and attaches to these receptors, acrosomal cap again releases some substances to digest this barrier. That means zona pellucida. What are those substances? Those substances are mainly acrosin, and other substances are esterases and neuraminidases. These substances will be released from the acrosomal cap and digest the, what is this barrier? Zona pellucida. Clear? Now this sperm reached here and attached to the vitelline membrane with the help of integrin peptides. Actually, integrin peptides will be present over the surface of oocyte as a receptor. So this sperm will go and attaches to the integrin peptides. Clear? So now the sperm is here. Now the sperm attached to the vitelline membrane. Clear? right? This fused part, actually there is a fusion of cell membrane of oocyte and the cell membrane of sperm. So this fused part will be broken down. This fused part will be broken down, right? Now third barrier also broken. That means third barrier also opened. Clear? Now as soon as it open, head along with tail enters into oocyte. Clear? So here there is no nucleus, there is no anterior centriole, and there is no axial filaments. Only cell membrane remains outside. That means it will not go along with cell membrane. It left the cell membrane outside and it will enter into cytoplasm of oocyte. Clear? Once sperm enters into oocyte, there will be formation of calcium wave. That means calcium wave sets throughout the cytoplasm of oocyte. When the calcium wave sets, there will be initiation of two important processes. What are those? First one is completion of second meiotic division. Actually, this is before entry. This oocyte is secondary oocyte. Is it? The secondary oocyte has to convert into mature oocyte. Clear? When it will happen? After completion of second meiotic division. Right? When it will initiate, as soon as sperm enter into oocyte, formation of calcium wave. That means calcium wave sets. That calcium wave setup initiates the completion of second meiotic division and production of mature oocyte, liberation of second polar body. Actually, within the perivitellin space, before fertilization itself, there was first polar body, right? Here, after completion of second meiotic division, there will be one more polar body. That polar body is second polar body. So here two polar bodies are present in the perivitellin space, right? That is one process. Another process is fusion of cortical granules. Cortical granules means what? At the periphery of oocyte, there will be some granules. These granules what we call cortical granules. These granules are cortical granules. These cortical granules fuses with the cell membrane. That means fuses with the vitelline membrane. Imagine here, this is fused with the vitelline membrane and releases the substances which are present in the granules. So they will release into it. Where they will release? They will release into the perivitellin space. What are those substances? Actually, they are hydrolytic enzymes. These hydrolytic enzymes will digest the ZP2 and ZP3 receptors. That means what? There is no receptors for the further sperms. Once one sperm enter into the oocyte, there is no entry for the further sperms. Why? Receptors are destroying. No receptors for the remaining sperms. Along with that, these substances will block the vitelline membrane. That means it will alters the permeability of vitelline membrane. It alters the permeability of vitelline membrane, which leads to prevention of entry of further sperms through the vitelline membrane. This process, what we call vitelline block. So, zonal reaction and vitelline block helps in the prevention of entry of polyspermy. Clear? So, That is about zonal reaction and vitelline block. Clear? Now this tail will be degenerated. Right? Here female nucleus and this is male nucleus. Male nucleus means nucleus of sperm and this is nucleus of ovum. Right? These nuclei will enlarge. These nuclei will enlarge and becomes male and female pronucleus. In the process of enlarging, there will be some important processes will happen within the nucleus of these gametes. Right? So what are those important steps? The nucleus of both gametes contains what? Chromosomes. Is it? How many chromosomes are present in the sperm? 23 chromosomes. Here 23 chromosomes. Clear? These chromosomes will decondense and undergo DNA replication. Why? Why it has to happen here now? See, let me draw some diagram. Here I am taking male nucleus, that means nucleus of sperm, and this is nucleus of ovum. Here 23 chromosomes are there, here 23 chromosomes are there. Once they unite, total it becomes 46 chromosomes, diploid number. These two are haploid and this is diploid number. Right? Once they fuse, resulting zygote contains 46 chromosomes. Right? This resulting zygote will undergo first cleavage division, cell division. At that time, if there is no DNA replication, again there will be a reduction division. Is it? To prevent that, there will be DNA replication. DNA replication. Actually, before DNA replication, chromosomes in the gametes will be like this. Is it? Chromosomes in the gametes will be like this. Is it? Once DNA replicated, chromosome structure becomes like this. That means double structure. You know already, one chromatid, one DNA. This chromatid, another DNA. So totally 2 DNAs are there. Further, DNA replication is needed. Once these nuclei completes their DNA replication, at the time they enlarge their size and becomes pronucleus. Now after completion of DNA, this is male pronucleus and this is female pronucleus. At this step, this oocyte, what we call ooted, at this time, that means cell contains 2 pronucleus, male and female pronucleus. At the time, this oocyte, what we are calling ooted. Clear? Now, here both nuclei will approximate with one another. So, they will come in the center. They will come in the center. Right? After that, there will be, what is this? What I am drawing here? Anterior centriole of sperm. Clear? Here, as soon as approximated with one another, destruction of or disappearance of nuclear envelope. Nuclear envelope will be disappeared, and this anterior centriole of sperm splits into 2, and these 2 migrated to the opposite pole. That means if it splits into 2, one half will go this pole, another half will go this pole. This pole. Right? Now these centrioles organize to produce microtubules For what? For the formation of aster or achromatic spindle or spindle. Clear? So for that these anterior centrioles are necessary. Clear? Now once this nuclear envelope disappears, what will happen? All these chromosomes which are present in the male pronucleus and female pronucleus arrange in the center of the cell, that means arrange in the equator plane of the cell and entangled within the aster or within the achromatic spindle which is formed by the centrioles. Clear? That arrangement I will draw here, and to avoid confusion I am removing these cortical granules. So these nuclei disappears their nuclear envelope, and arrangement of chromosomes. Which chromosomes are these? Both maternal and paternal chromosomes. Imagine this is maternal chromosome and this is paternal chromosome, and here again paternal chromosome, here maternal chromosome, right? All these chromosomes arranged in the center, in the equator plane, right, and entangled within the aster. That we know in the cell division we have discussed. These kinetochore microtubules from the aster, they go and attaches to kinetochore of chromosomes, right? So these are kinetochore microtubules, and here aster microtubules are anchoring microtubules, and which contain some, what are these microtubules? Polar microtubules. Clear? So now both maternal and paternal chromosomes are entangled in the achromatic spindle, right? So this is at the metaphase of first mitotic division of embryo or zygote, right? This is first mitotic division, right? According to our definition, we completed fertilization. What is our definition? It starts with fusion of male and female gametes, ends with intermingling of maternal and paternal chromosomes at the metaphase of first mitotic division of zygote. So this is the thing. Clear? So from here, starting of first cleavage division. So cleavage is separate topic, so we will discuss there. Up to here fertilization. Clear? Now we have completed fertilization discussion. Then last heading, effects of fertilization. Effects of fertilization. Actually that effects we have already discussed, but still I will tell you for name shake. I am not writing, just you write it down. I will tell very slowly. First effect is immediately after entry of sperm, what will happen? There will be completion of second meiotic division. First one is completion of second meiotic division and production of mature ovum and second polar body. This is first effect. Is it? It was happened. Then second one, restoration of diploid number of chromosomes, that I have discussed here. Here 23 chromosomes, here 23 chromosomes. Both male and female pronucleus united. Totally it became 46 chromosomes. So from haploid to diploid. Clear? So restoration of diploid number of chromosomes. Second one, write it down. Restoration of diploid number of chromosomes. Then third one is determination of chromosomal sex. How come? See here, Here 23 chromosomes are there. This is male pronucleus. In this male pronucleus, if the 23 chromosomes, within this 23 chromosomes, 22 autosomes, and here one sex chromosome, imagine this is X chromosome. Of course, in female gamete always 22 autosomes, sex chromosome is X chromosome only, right? When these two pronucleus united, resulting zygote is 44 autosomes with XX. So resulting zygote is female. If sperm contains 22 autosomes with one Y chromosome as sex chromosome, at the time here 44 X and Y. So resulting zygote is male. So male gamete will decide the sex. Male gamete will decide the sex, right? So this is determination of chromosomal sex. Clear? Then another one is, I think fourth one is activation of metabolic activating factor, which leads to initiation of cleavage. Activation of metabolic activating factor. Metabolic activating factor which leads to initiation of cleavage. Was it happen here? Yes. DNA replication has been completed and all the chromosomes are arranged in the equator plane and they are ready for the division. So initiation of cleavage by activation of metabolic activating factor, right? So that is another effect. Then another one is prevent polyspermy. Prevent polyspermy. How it will prevent, that we know. Zonal reaction and vitelline block. This ZP2, ZP3 receptors will be destroyed, that means digested. So no entry for the further sperms. Clear? So prevent polyspermy. Then another one, final one is formation of unique zygote, genetically unique zygote. How come? See here, 46 chromosomes are there. These 46 chromosomes are not identical to father and they are not identical to mother, because they came from both maternal and paternal chromosomes, right? So this is totally not identical to mother and totally not identical to father. It is genetically unique zygote formed after fertilization, right? So these are the effects of fertilization. Remember carefully, this is very important short note question. Clear? So with this we completed discussion about fertilization. Next session we will discuss another topic.